Hello everyone, this is Tushar Biswas and welcome to the Study Companion. Today we are going to learn about the Griffith experiment, the experiment which solely helped the humankind in understanding that there are various factors that are being transferred from one organism to to another organism, which is changing this new organism in the form of the previous organism. So that there is something is being transferred from one organism to the other, which is resulting in the change in the new organism. So without any further delay, let's begin. Okay, the Griffith experiment. Okay, so everyone, before speaking of the Griffiths experiment, I am going to go into a bit more details of what happened, what inspired such an idea. First came our very own and beloved Sir Mendel, who gave us the idea of the factors being passed from one organism to its progeny to another organism that is belonging to its progeny. He gave us the basic idea of inheritance. Then came Sutton and Boveri, who with their experiments gave us the chromosomal theory of inheritance. Okay, first comes Gregor Mendel, Sir Gregor Mendel. Then comes Sutton and Boveri. Through their experiment, Sir Gregor Mendel, he was a monk. He was a monk actually. He gave us the chromosome. He gave us the theory of inheritance. Okay, Sutton and Boveri with their experiments gave us the chromosomal theory. Chromosomal theory of inheritance. So after Sutton and Boveri, we get Thomas Hunt Morgan. Mr. Thomas Hunt T.H. Morgan. What did he do? He gave us the idea that chromosomes had some genes. These genes were the ones that were being transferred from one progeny to the other. So he gave us the idea of the transfer of genes in chromosomes. Okay. So I was checking whether my system is recording it or not. There was a technical glitch over here, hence I couldn't upload any videos in the past few weeks. Okay. So chromosomal theory of inheritance. Right now we have Thomas and Morgan. He said that genetic traits are he said traits. What did he say was traits were a part of genes that were situated on chromosomes. So chromosomes carried genetic traits. Chromosome carried traits which were genetic in nature. Okay, Thomas said that genetic traits are present on chromosomes. So what did Thomas say? Thomas said character traits are genetic in nature and they are present on chromosomes. So what did he say was traits? Genetic traits were present on chromosomes. Traits present in chromosomes were genetic in nature. Okay, this in itself has two explanations. Traits are present on chromosomes and are genetic in nature, or genetic traits are present on chromosomes That is also one kind of explanation to it. Okay, so everyone, T.H. Morgan said that the genetic traits were present on the chromosomes. Genetic traits were a part of the chromosomes. Okay, then comes our very own Mr. Frederick Griffith. After T.H. Morgan, after Thomas Hunt Morgan, we have our very own scientist, Sir Frederick Griffith, who in 1928 had postulated a theory. Which theory I'll tell you in the conclusion. Okay, in the conclusion of this very video, this very topic. So he was, now let us focus our whole story on this. So I hope everyone understood that Mr. Gregor Mendel, Gregor Mendel gave us the theory of gave us the idea of inheritance. Then Sutton and Boveri through their experiment gave us the chromosomal theory of inheritance. Okay, if you want, I can dedicate a video to this part. Okay, and then comes T.H. Morgan, who gave us the idea that traits are genetic in nature and they are carried from one person to the other in the form of chromosome. They are present on chromosome, and hence these traits can be carried. He didn't say that they were genetic. Okay, the genes, what were they made of? Now that was the basic question. He said that they were genetic. Genes idea came from this in inheritance. Then they also gave us the idea that chromosomes were present, and on that the genes are present. Then Thomas Hunt Morgan, on his work on fruit flies, he did some researches on fruit flies, and he saw that genetic traits, okay, the genetic traits, if this is a chromosome, there might be a space over here, there might be a place over here, a place over here, and here. So these genetic traits, some may be linked to each other, some might not be linked to each other. So what happens is that the ones which are linked can be transferred from parents to the offspring easily. So sometimes what you tend to see, is that the parents' characteristics are being resembled in the child. Okay, the child is resembling the parental characteristics. How is it happening? Because there are some genes which are linked to each other. Okay, the genes which are linked to each other, hence these genes are being transferred from the parent to the offspring. Okay, T.H. Morgan said that the parent's genes on a chromosome are sometimes linked, sometimes non-linked. So the characters which are linked to each other are being transferred in that specific set only. The linked characters, the characters holding their hands, they're pulling each other. Come on, let's go. So they are taking them to the offspring. They're taking them to the offspring. Hence, the parental characteristics are being completely uh, dominated over there, completely showing off over there. Okay, in the progeny. I will next time I will make a video on this, Mr. T.H. Morgan's work on fruit fly. We'll come to it. First, let us focus on the easier ones. Then we'll come to the more difficult parts. Because you need an idea on this before you go focus on this. Okay. Linkage and recombination, well, I'll come to that part. I would surely teach you this. Right now, I won't focus over it. Just know that there are some traits. All the traits are present on chromosome. Number one observation. All traits are present in chromosomes. Number two observation. Each and every trait is being encoded by a gene. Number three is that this gene that is coding for these genes are present on the locuses of chromosomes, homologous chromosomes. So there might be a stuff where two genes are holding each other's hands, are linked to each other. Hence they are being carried forward to the parent, to the offspring. Means if the parent has a habit, if the parent's characteristic was tall and handsome, a child will also be. And for example, if one of the parent was tall and handsome and the other parent was short and were rather non-handsome, or I wouldn't use the word ugly, non-handsome, not that much beautiful, tall and beautiful, tall and not beautiful, something like that. The child, if the child is tall, the child will be handsome or beautiful. If the child is short, will not, that child will be short or short and not beautiful, non-beautiful, because either one is being generated. So we now know that genes not only segregate like 100% independent of each other, but sometimes rules given by Mr. Mendel is not at all in par. Okay, sometimes genes take with them what is their, what you say, like best friends. Okay, best friends. So this might be a gene for tallness, this might be for beauty or handsomeness. So what do? When this gene goes anywhere, this one follows. So if the child is tall, the child is bound to be good looking. If the child is dwarf, according to the other parent, the child is bound to be non good looking. Okay, I hope I made myself clear regarding T.H. Morgan's work. T.H. Morgan's work was in T.H. Morgan's work was in 1910. Okay, he published that in 1910. Not actually 10, I would say. It rather took him many years. It rather took him many years. 1908 to 11. 8 to 11 he took, and he published his work. Then in 1928, our Frederick Griffith, Mr. Frederick Griffith, did this kind of experiment. Okay, Frederick Griffith did this kind of experiment, which we'll talk about right now. Let me erase the board. So everyone, I just had paused my video to just clear off the board, and right now let us focus, or else the time would be wasted. Okay, so let us focus on this part. I think the board has gone a bit backward. Yeah, it is okay now. Now we'll be speaking of Frederick Griffith. Frederick Griffith. It was in 1928 when he did this, when he came out with the conclusion. Prior to this, he was working with pneumococcal strains. Okay, pneumococcal strains. What pneumococcal, like, what were they actually? They were this bacteria for pneumonia. Okay, Streptococcus pneumoniae. Pneumonia, you can also say like that. Streptococcus pneumoniae. He was working on Streptococcus pneumoniae, and this was causing pneumonia. And he took two strains. He was working on two strains of it. Okay, two strains of it. Now strains are just very small differences within the same species. Okay, the genus, the species, the what is it? Genus, the species, the subspecies can also be different, but at the most basic level, there's a difference between strain. Okay, there might be a bacteria, might be same, might be in the subspecies also. Okay, two bacteria taken from one single subspecies might vary very slightly in the physical terms, in the physical characteristics. Hence, then we come with the idea of strains. Okay, types of bacteria present in a species, subspecies are known as strains. Okay, difference in bacteria or microbes, differences in microbes, differences in microbes present within a genus, species, or subspecies that differ with each other only by some physical means or only by some physical means or or genetic makeup, genetic makeup as in small differences or small variations. Okay, genetic makeup, that is small differences, or not differences, small variations amongst organisms belonging to uh, belonging to the same genus, species, or subspecies. This is what strain definition of strain is. Is my camera recording? Yeah, it is recording. Differences in microbes present within a genus, species, or subspecies that differ among each other only by some physical means or genetic makeup. That is, small variations amongst organisms belonging to the same genus, species, or subspecies is known as are known to be different strains. I hope everyone has copied it down. Please copy this one down. Okay, please copy this one down. I'll be pausing the video and just erasing off this part only. Only there is a small genetic difference. There is small variations in the species. Hence, their body parts, the body composition might vary a very little, and hence the term comes in as strains. Strains are same bacteria, same what do you call same microbes when they differ with each other just by some small bodily characteristics, and we know bodily characteristics comes from our very own genetic material. So what happens is that we have a small variation, and due to that variation people look different, right? We people, as in we humans, look different. We have different sets of characteristics. Why? Because variation is there, so we might be also known as strains. But for bacteria, they're very small. The physical characteristics are important for us to know. So the based on the physical characteristics, we differ bacteria belonging to the same genus, species, and subspecies as strains. Small physical difference, strain. We actually don't as much focus on genus and species. We might focus on species or subspecies the most. Genus is generally not preferred, but still it is included in the definition in case we might focus on it. That's very rare focus on genus. Majority focuses on species and subspecies. Okay. Majority is for species or subspecies. Genus is just for the sake of writing. or we might as well remove genus, okay, because physical differences in genus causes the formation of species. There are high physical differences, not genus. Let's remove genus. Yep, let's remove genus. Don't write genus. Remove genus. Species or subspecies? Okay, species or subspecies. See, the species is same, the strain will be different. So species or subspecies. Generally remove the genus and copy the definition down. And right now I'll be pausing it and erasing my board. I have erased my board, and he took two strains. One strain was the S strain or the smooth strain. S stands for smooth. And the other one was the R strain. R stands for rough. Now why smooth and rough? Like based on what? Based on what was the differences? The smooth strain had a complete, if this was the bacteria, if this was the bacteria, there was a complete and uniform bacterial coating or covering, bacterial cover or coating, and this coating was formed of none other than our very own polysaccharide. Multiple carbon sugar is known as polysaccharide, right? So this is polysaccharide. Okay, I'll bring the board a bit up front. I guess it's not legible. Yeah, it's legible right now. Okay, I'll just set it up a bit. Now, polysaccharide. So there was a complete coating of a polysaccharide, and for the R strain, the same bacteria did not have a complete coating. It had bits and parts of it, which made it rough. See, it made it rough. This was the open area. This was an open area. There was coating over here, polysaccharide. These areas which are uncoated, where the virus, where the bacteria was seen. When these bacterias, S strain and the R strain, entered the body systems, the body defense mechanism, the immune systems, the immunity came out, rushed, and when they saw this bacteria, they found that this is a polysaccharide, this is a sugar, if this is a sugar, this might be a digested particle, this might be a sugar in nature, this cannot be anything more. It did not understand that there was bacteria hiding inside the sugar covering. It came out, it saw that this is sugar, this is a sugar coming in, so it is not potentially hazardous to us. Okay, so just a sugar molecule will get digested, it's actually good for us. It never knew that there was a bacteria inside. But when R strain came in and the immunities rushed towards the R strain, they saw that there might be sugar over here, but we can also see that bits and parts of a bacteria is poking out of the sugar. Okay, bacteria is being poking out of the sugar. If in the first case the bacteria was covered by the sugar like this, so when the molecules came in, they saw this part, this is polysaccharide, everywhere they saw was a polysaccharide. Okay, but in the case of R strain, it was like this, bits and parts were coming out of bits and parts of the bacteria was coming out of the covering. Hence when the immune cells came in, they saw this sugar, but they also saw the bacterial parts. See the bacterial parts. Hence, they understood that the bacteria was trying to camouflage itself. Now, what did they do? They just killed this bacteria. Hence, the R strain was easily killed. It was recognized, that's the reason it was killed. This one, on the other hand, was able to mask itself and taking the cover of being a polysaccharide, taking the cover of being a sugar molecule, taking a cover of being a sugar molecule, the bacteria rushed in and caused its infection because the body wasn't able to recognize that it was a bacteria in disguise. Okay, so now I'll just again pause it and just erase the board. So I have wiped it off. Now let us see the actual experiment. What did he do? He took 4 cases. Case 1, case 1, case 2, case 3. Okay. First case, he took the S strain bacteria, injected it inside a mouse or mice, what do you say, and saw after what happened to it after some time, and it was found that the mouse had died because the infection had established. Okay. Next up, he took the R strain. He took the R strain and again injected it inside the mouse. Then the mouse did not die. Mouse was alive. Mouse was alive. The Okay, I guess it's visible right now. Yes, it is. So the mouse was now alive. Mouse alive. Yep, it's visible. Yeah. So guys, this I had paused the video once. I just refilled the marker and I just erased this slide because I had to explain something. Now, heat killing a bacteria. If a bacteria is present and you heat kill it. To kill a bacteria, you need to do 2 things. Stop the metabolism, then break down the genetic material. Okay, there are 2 things to kill a bacteria, not 2 ways. Okay, the first method is stop metabolism, means when the bacteria is getting too much of heat, the proteins are being denatured. Biomolecules class 11. Proteins survive at a specific heat and specific temperature and pH. When the temperature rises, the proteins denature, so does the enzymes. What happens is that the metabolism stops. The digestion and the formation of energy from the food particles digested is known as metabolism. Okay, this metabolism then stops. It will stop because the enzymes have stopped functioning, but the bacteria is not dead. Bacteria goes into dormancy. What happens is that the genetic material is the main thing. If you are not able to break this, the bacteria is partially dead. Okay, the denaturing of the proteins and enzymes might stop the food production in bacteria. The food utilization and the production, the metabolites production of the bacteria might stop. But the genetic material is alive. If it's transferred to one of the bacteria, it will again live. It will again help the other bacteria in mutation. That's how mutation occurs. I will come to it, not in this not on this video, a bit later on. Okay, so this is how to kill a bacteria. So after this, how do you understand the bacteria is partially killed? Metabolism produces metabolites, produces products products are metabolites. So if you do not find metabolites around the bacterial colony, you would understand the bacteria might be dead. So that is what you known as is partially killed. Okay, partially killing of a bacteria, partially killed. Okay, to kill a bacteria, you first need to heat it. You continuously need to heat it. The first amount of heats will denature the proteins, the enzymes, and the metabolization will stop. Metabolization is stopping, hence you will not find metabolites, the products, around the bacterial colony. That's first step for you, but still you need to heat. If you stop over there, the bacteria is still half living, because the genetic material is living, the genetic material is intact. Now you continuously keep heating, and at the and any heat more than 90 degrees Celsius will denature this genetic material. When this genetic material is destroyed, the bacteria is completely dead. Prior to this, only this part, only after doing this part, it is only partially killed. Okay, not completely killed. I hope I made myself clear. Erasing this part. So what did he do after finding out that thing? He actually didn't find out that thing back then. It was considered, since we human beings, when we die, our metabolization stops. We stop excreting our waste products and everything. So he thought that the bacteria might behave in the same way. So the stopping of a metabolization, for stopping of the formation of metabolites around the bacterial colony, might be might prove the fact that the bacteria have died. So what did he saw was that he took a bacterial plate where the bacteria were present, and first, after, before heating, the bacteria produced 20 micrograms of metabolites, and after 20 minutes, these bacteria produced 50 micrograms. Okay, but after heating, this is 20 micrograms. So this is our benchmark. This is our benchmark. So he took two plates. Where the benchmark was the same, amount of bacteria was same, and he saw from 20 micrograms after 20 minutes. This was non-heated. This was heated. After 20 grams, first the 20 grams was measured, then it was not heated, this was heated, the plate was heated, bacteria were subjected to temperature change, and he found that the 20 grams have not increased, 20 micrograms have not increased. Hence there was no formation of any more amount of metabolites. That were formed in the first few phases was 20 micrograms, and after that, when the heat started, no new metabolites were formed. The one which was present was there. The one which was to be formed wasn't even formed. By this time, within this same time, it might have risen up till 50 micrograms, but it is 20. So metabolization has stopped. Metabolization has stopped means the bacteria might be dead. According, like what happens with the humans, metabolization stopped, bacteria might be dead. Then he took the S cell. I'll be erasing the board, so I'll pause it. So guys, I had again paused the video to just erase the thing. Then he did two other things. First he took heat killed S strain. According to him, heat killed, but he never knew at that time that they were actually partially heat killed. Partially heat killed S strain. Okay, he took them and injected in the mouse, and to his surprise, the mouse survived, or the mouse was seen alive. Okay, the mouse survived, a mouse was alive, whatever you like to say. Then he took just because, why? Because when the heat was there, this very thing is had happened was that when the heat was there, when it was being heated, these polysaccharide areas had dissolved, and the bacterial areas had come into limelight. So it looked like the rough strain now, with parts of the bacteria emerging out of the coating. Hence the body immune cells easily caught hold of this bacteria in disguise and killed it. So the infection was not caused, and the my mouse survived. Then he took an R strain. Then he took the R strain and added this whole thing, partially heat killed S strain. Okay, he took them and he injected in the mouse, and he expected it to not die, to survive. But he, according to him, and now we know why did this happen. But he got surprised by the outcome. Mouse had died. The mouse had died. What had happened? So out of from these four experiments, the R strain looked somewhat like this. Okay, with bits and pieces emerging as a cover, whereas the other parts emerging out of the cell cover, hence they were killed. Over here this thing was completely covered, hence it was mistook as a sugar. This thing again, it was like this previously. When heated, these melted away, and but it started looking like an R strain. Now R strain bacteria, parts were coming out of this thing. These melted and it resembled R strain now. But when both of these stuff came together, this was the heat killed S and this was the R strain. Okay, these were the R strain, R and S strain. Both of them could be recognized by the body, but they were able to establish the infection, and hence the mouse died. What did happen? What did happen? What was his conclusion accordingly? So I'll just erase the board and get back to you. I have erased the board, and time for the conclusion he derived. What did he conclude? What did he conclude? So his conclusion was that there is or are presence of some factors that have the ability of changing one strain of organism, changing one organism belonging to a strain changing one organism belonging to a strain to another new organism belonging to another strain. So there was a factor, there was a there was a presence of factors who had the ability to transfer one organism into another organism, transform. So what was happening was one strain organism belonging to one strain was being transferred to a new organism belonging to another strain. So what was his idea was that the partially heat killed a strain bacteria was transferring R strain bacteria to live S strain bacteria. So partially heat killed S strain. Right now we know that this S strain had that sugar covering just encoded in their genes. R strain had this kind of sugar covering, had this kind of bits and pieces of sugar covering encoded in their genes. So what was happening was that actually this gene had flowed in over here. The living R strain, when coming in contact with the living DNA of the S strain, had undergone mutation, and this R strain has become S strain bacteria now. This was living, metabolization live over here, metabolization live, DNA live, metabolization dead, but DNA live. What was happening over here was that the DNA, though living, was not enough to help the bacteria survive because the covering wasn't formed properly. But the DNA in this case was enough to keep the bacteria living because it had generated a cover around itself. But the metabolization was dead, hence the bacteria couldn't function anymore. What was happening was that since the metabolization had died, the DNA was transferred to this. Over here, this new DNA came in contact with this DNA. They again came together. They formed a new DNA where codes of forming this thing was present. After forming this thing, this R strain had the metabolization live. It was living, so the metabolization was there, which was exactly required for this DNA. So new DNA was being formed. This DNA carried codes to form this thing. Now this rDNA, not rDNA, this R strain would look like this. The progeny, the ones following this thing would look like this because this new DNA had come in over there and formed a new DNA in presence of a living cell. This DNA had come into limelight. They got the ability to do work and produce a new offspring. This offspring was the S strain bacteria, though it was later on. No, this thing came out later on. His observation was this and only this, that there are some factors that have the ability of transferring one organism to the other organism, one strain into another strain. Okay, transferring, not changing. Changing or transferring. Transferring is a better term. Transferring. Transferring. Okay, so till here. This was all of Griffith's experiment. Next one we'll bring Avery McLeod McCarthy experiment, which is the rectification of this experiment only. And until that, stay tuned, keep studying, and keep watching the Study Companion. This is Tushar Biswas signing off.